much. Well, many thanks for inviting me virtually to this meeting. Well, you invited me physically, but I'm coughing because I'm recovering from COVID, so I better stay away from you. And I've been asked to talk about what is happening regarding Duchenne muscular dystrophy and antisense oligonucleotides. I work at the Institute of Child Health. I am a pediatric neurologist. I'm involved in a number of clinical trials. I don't have any direct gaining from any company or any product apart from taking part in some advisory board, but I don't have any particular association with one particular company. And I will take you through these different points. Briefly, I will not go into detail of the past, but where are we in terms of the recent data on molecular efficacy and clinical outcome in current generation morpholino antisense oligonucleotide, PMOs. I will present some ongoing work in my group in terms of trying to understand variability of response. There is clear work for next generation antisense oligonucleotide, both in terms of peptide conjugated PMOs, but also transferrin receptor PMOs. And I will give a brief update where this field is, but also I will finish with some of the ongoing work in our unit and in collaboration actually with multiple people regarding using antisense to restore dystrophin in brain. And you will hopefully understand why we are interested in that. So this is my only slide in Duchenne muscular dystrophy because it doesn't matter to go into the fine detail, but the point, I suppose, is that this is a common disorder for childhood muscular dystrophy. Approximately 100 children are born every year in this country. Is due to mutation in this very big gene that is big enough that is a challenge to put it back entirely by AAV gene therapy. And therefore, alternative strategies to try to deal with the mutation have been devised. Now, mutation, these are the 79 exon of the dystrophin gene. Mutation are mostly out of frame deletions. And the reading frame is indicating different color code. And in Duchenne, sometimes and often 1 or 2 or 3 exons are missing, and this causes the inability to produce this protein that normally will be located at the periphery of every single muscle fibers. There is an experiment in nature where patients with a milder form of muscular dystrophy called Becker muscular dystrophy also have deletions, but this time are in frame. They can produce some protein in the muscle, and although it's better not to have Becker because it's still a progressive disorder, and maybe a third of the individuals may end up using mobility aids later on in life, but clearly it's much better than Duchenne. So the idea is how can we exploit this information for with splice switching antisense? And just to make the story, the past story shorter, 4 drugs have been approved by FDA based on a surrogate endpoint. And the first one was an antisense to target exon 51 at epilsin. And then, of course, depending on where the mutation is, you need to skip different exons. Then targeting exon 53 with golodirsen and viltolarsen have been approved by FDA. And more recently, targeting exon 45 with casimersen was also approved. And I will not go back in the, if like in the work of the last decade on this morpholino. I will just present some of the more recently published data these are data that come from Sarepta presentation. This is a very recent presentation of a larger clinical studies that where the level of dystrophin, both in terms of percentage of dystrophin positive fibers, intensity with semi-quantitative digital capturing of dystrophin at the sarcolemma, and Western blot were present. And if you like, it is clear that this again, and these values are the difference between pre and post, okay? So this indicates that there is exon skipping, that in actual RNA wasn't in this table, but there is exon skipping, there is some protein, the level are very small. No question about that, less than 1% in this particular example. This is work that actually my group has published very recently with collaboration with Sarepta and a European grant. We did a phase 1, phase 2 study where we targeted exon 53 with, and I didn't say this, but these are morpholin antisenses given intravenously every week to patients. And we were able to demonstrate after 48 weeks that there was both at the RNA level and at the protein level and at the level of dystrophin positive fibers, production of dystrophin. I think this was unequivocal. Again, the level in the range of approximately 1% in mean, with a variability in some patients up to 3–4%, actually even more, but some patients having just traces. And then very similar data in terms of the level of dystrophin production were demonstrated by this time Sarepta with targeting exon 45. Now, these are the level of dystrophin at week 48. I think that you may think this is a much more because it's the double, let's say, of the exon 51. But I think an important aspect for you to be aware of is that the baseline in patients with this particular mutation is higher. These patients don't have virtually zero as we have in our study, but they have nearly 1% to start with. So I think that's an important aspect. So again, there is dystrophin production, but don't be fooled by just the number. You just need to look into the baseline. Very similar data have been produced this time by NS Pharma. There have been 2 studies that have been published, one by a group in the States, in German Neurology just last year, where 2 different doses have been used. I did not say, but the previous data were at 30 milligram per kilo of the Sarepta drug, while the NS Pharma had tried dose up to 80 milligram per kilo. And this is the percentage of dystrophin on mass spec and on Western blot that were identified in the American part of the study. This is the level that were published in the Japanese part of the study, the same drug, same doses. And again, if like a sense that higher dose are better than lower dose, but again, some variability between the 2 studies that probably reflect more than anything else, relatively small number of patients, but unequivocal production of dystrophin. And sense from both studies that 80 milligram is better than 40 milligram. Now, the question is, does it matter in terms of clinical efficacy? I think it does, but don't expect miracles from relatively small level of dystrophin. So again, I'm only concentrating on very recently published data. I'm not going back in previously published data you can find in PubMed. This is a paper that has been published just last year. That is a cumulative data from Sarepta on patients who have received the eteplirsen, so skipping exon 51, across a number of different studies. And on the top is compared to, these are studies that are going on now for years, and therefore the open label extension studies are compared to untreated patients in a number of different registries, actually in this particular case, against the Synergy American registry. And as these are all American patients, it's probably not completely wrong. And they were able to demonstrate this is, gross percentage of patients losing the ability to walk. And you can see that after probably 2 years, 2 to 3 years, you start to have a divergence of the curve. And after 6–7 years of treatment, there are still approximately 25% of the patients receiving the drug being on their feet, while 0% of the patient would be still able to walk at that time. And again, there is some sense that respiratory function is protected, and that the decline, the year-on-year decline in the different study is reduced. And if you compare to the untreated patient, in whom the loss of respiratory function is 6% every year, the treated patient lost approximately half per year. In our own Golodirsen clinical trial, this is work that we have published just a few weeks ago. We were able to demonstrate no divergence between treated and untreated patients after 2 years, but a trend was, in all honesty, non-significant, but I think there was separation of this trend of these two curves at 3 years, and separation of the number of patients who lost the ability to walk. Again, this is a small study. We only had 25 patients in treatment. So again, you can see the p-values are non-significant, but there appears to be a beginning of a separation of the two curves. And similar data on the respiratory function, again, suggesting very similar to eteplirsen, approximately half of the decline in the respiratory function in the treated patient. In the data from Wiltor Larsen, at the moment, we only have data available from, if like, and these data suggest that there appear to be an effect in the already at week 25, although in the Japanese study, this is the American study, in the Japanese study, there was no difference between the high and low dose, and considering the difference in dystrophin, I think that more time will allow to have better separation from natural history studies. In terms of this is now only exon 53, so the work we have done with Golodirsen, dwelling a little deeper from a molecular perspective, I suppose, we were able first to demonstrate between baseline and week 48 that not only there is some dystrophin produced in the patient, and you can probably not appreciate very well here, but in our paper, we demonstrated that a significant increase in the protein of the dystrophin complex. So it's not only dystrophin, but also the associated protein that are restored. And I think one thing that I was very interested was, again, if you concentrate on the bottom part of this panel, this is this time looking at how much damage is in the muscle, okay? So this is the baseline. These fibers that lit up here as individual fibers are fibers with markers of degeneration and regeneration. And we were able to compare the pre-treatment to the post-treatment. And we were able to demonstrate that in the majority of patients, there was the green bar suggests that there were less degenerating and regenerating fibers at 48 weeks compared to before. It wasn't in all patients. Some patients didn't had still a significant number of regeneration, but then we were able to demonstrate that those patients who had the higher level of dystrophin, those were the patients in whom the regeneration was lower. So I think, again, some indication, apart from clinical aspect, that there may be, or there appears to be a molecular pathological effect at the muscle level. We have characterized a little further two aspects of the response to therapeutic intervention. The first is on transcript imbalance. The second is why there are differences. You know, in this slide here, I demonstrated there is difference, and there is difference both in terms of degeneration, regeneration, and level of dystrophin. So in terms of transcript imbalance, for those of you who are not familiar with the dystrophin gene, this is a pretty, it is a very large gene, and there appears to be particularly in patients, particularly affected by imbalance of transcript between the 5' end of the gene and the 3' end of the gene. And this is not necessary for all big genes. There is already some transcript imbalance in the untreated patient, and in the treated, in the control, but in the patient, the transcript imbalance is very dramatic. And of course, there is an issue that I will touch upon that is slightly misleading when you read the level of efficacy of targeting the MDX mouse, because the MDX mouse has a mutation at the very beginning of the gene. So the level of RNA in mice are very different from what we find in patients, because in patients, this is the area we target, okay? So that's an important consideration, because our patient, compared to mice, have less RNA to start with. But then we were able to look into the transcript imbalance in our patient using skin biopsy obtained from them and with myogenic transformation of these cells, and looking with the fluid DMD card, the entire transcript in this patient. And interestingly, we were able to demonstrate that the treatment does improve, not in all. So 0 means that there would have been no effect on transcript imbalance. Positive means that there is a positive effect on the transcript imbalance, meaning with partial restoration of the, level of transcript across the entire gene. So I think that is something of interest. The other thing we have done using the same system is firstly to compare the myogenic cells, and these are the various patients that have been studied, to the muscle. And we were able to demonstrate, and I don't have it here, so you need to take my words for it, that actually the myogenic culture protein level very well compared with what we did find in the muscle of the patient. And again, you will appreciate there is variability. And the variability, therefore, appears to be intrinsic to that particular patient. Maybe we couldn't target very well muscle in vivo. And we are currently, this is ongoing, we're currently looking more in detail using base score at the level of transcript, both unskipped and skipped transcript in patients with different deletion. And it looks as the limiting factor is the fact that in the probably not totally unexpected in the low responder, the amount of also unskipped product, the amount of source RNA is very low, and there appears to be some genotypes where this source RNA problem is a bigger one. But I think this is again ongoing work. In terms of the, this is where we are in terms of the current generation antisense. There is, I think that the good news is that the PMO are well tolerated, but of course they require weekly infusion. I indicated that modest level of dystrophin can be produced. In my experience, it takes several years before a treatment effect becomes visible, at least in the patient I've been involved with. The second generation antisense are those where at the time it's been linked to the PMO, and Sarepta has an ongoing phase 1 study, but also where a receptor-mediated antisense uptake via the transferrin receptor is being exploited by 2 companies, Dyne Therapeutics and Avidity. So regarding the peptide conjugated PMO, this is a study that is ongoing from Sarepta, where etepilsen and the new drug have been compared at a different time point. In this particular case, at week 12, at week 24, in patient receiving etepilsen and the new PPMO. And on the left-hand side is the example of the difference between the level of skipping demonstrated at this time point and this time point. And this is another example where this time in the patient treated, this is the level of exon skipping that could be identified in the Duchenne patient treated so far. And this is the level of protein expression. And again, these are comparing the difference between the patient that, as we indicated in the past, receiving etepilsen and have not even 1%, while in the patient treated in the clinical, in this first 4 patients treated in the clinical trial, at 30 milligram per kilo, that is the optimal dose according to Sarepta, the level of dystrophin are considerably higher. They go at 6%, 7%, and Sarepta anticipated that chronic treatment may arrive to at level of at least 10%. I think is not free lunch. There had been a FDA hold because of a reversible hypomagnesemia problem observed in several patients. The company has now a strategy to deal with this, and the recruitment is started again for this study. So this is very early days. I think even a little behind is the Dyne Therapeutics and Avidity attempt to address the same problem, I suppose, by this time exploiting the transferrin receptor. And this is work that probably the first of the company to hit the clinical ground will be Dyne Therapeutics, with possibility of first patient to be treated this summer, but Avidity should follow very shortly after. And these studies are on cell culture. Sorry, this is, I apologize, this is a non-human primate, but a wild-type non-human primate with a level of exon skipping observed in the quadriceps, in the diaphragm, and in the heart. You can see this value by yourself. And this is, this time in the MDX mouse, the level of dystrophin obtained after a single dose of the equivalent of exon 23 transferrin receptor linked PMO, with a very respectable level of skipping and protein production in each of these tissues that are indicated here. Just to finish in the next 5 minutes is, this is more my pet interest. Coming back to dystrophin, I discuss about dystrophin. I told you that in muscle, the main isoform of dystrophin is absent because of mutation in these 79 exons. What I failed to tell you is that this isoform is also present in brain, but in addition, the gene for dystrophin encodes a number of shorter isoforms generated by internal promoters, and all the one in red are expressed in brain. In particularly important at DP140, DP71, but also the muscle isoform is expressed in brain. Now, the absence of dystrophin will lead in 50% of the patient to neurobehavioral problem and or cognitive problems. And I'm not going to give a detailed explanation of the brain phenotype. But one particular important and interesting brain phenotype, not originally described by these colleagues in Japan, relate to this unconditional fear response with the mice that after a short handling have paralyzing problem with stress, and they don't move. This is the curve of a wild-type mouse that after 15 seconds of holding is put in the cage. This is how it moves. This is the MDX mouse, but this is not related to the muscle side. And on the left-hand side, you have the MDX mouse, and on the right-hand side, you have an MDX mouse that has been treated with central administration of antisense oligonucleotides, because their central recovery of dystrophin, both in this study from Luis Garcia and Aurélie, and in the study from the colleagues in Japan, was able to restore some of the synaptic dysfunction that absence of dystrophin causes. So this is quite interesting. And so I was lucky, I suppose, to get before the major next mess of Brexit, to get a European grant to address these both clinical aspect and preclinical aspect. And I'm just very pleased to say that the progress is going very well. And there is a number of colleagues here on the right-hand side. You see Aurélie, Cyril, Yoshi, Annemieke, Federica, Vincent, and Manuela, amongst many others. And we are looking at the behavioral feature in mouse model, in MDX mice model that are deficient of the different isoforms, and we restore dystrophin using different antisense oligonucleotide and delivery methods. And the interesting thing is that we have mouse model where you can restore either the full-length isoform. So we have a mouse model that is deleted in exon 52. So if you skip exon 51, you restore, as in muscle, the main isoform, but you kill the one particular isoform that is called DP140, because the start site of DP140 is in exon 51, while if you skip exon 53, you can restore both isoforms. So we have ongoing work, and I'm only going to say that we have ongoing work where we are measuring a number of phenotypes, in particular the conditional fear memory, and we're able to demonstrate the improvement of this phenotype after brain dystrophin restoration. And recently, Yoshi was able to demonstrate that also skipping the exon 53 and therefore inducing production of DP140 was able to improve the phenotype in this particular new mouse model. So this is ongoing work. I don't think I'm giving justice, but it's only to say that maybe learning from what we're doing for muscle, also for brain, both for PMO, but also for all these different antisense oligonucleotides. So this is my last slide, is to say that I hope that, I think I am convinced that there is effect of the current generation antisense, but clearly this is the basis for getting next generation antisense developed for better activity. And I think we're clearly far from the cure for Duchenne. I think one thing that I didn't discuss, but of course is relevant, many of the studies that in my own study, the one I showed you, the divergence of the clinical trajectory after 3 years, well, the mean age of recruitment of this patient was 7.5. This patient had lost already 40% of muscle by the time I recruit them. So one, eventually treating much earlier children will probably be more rewarding for any of these antisense. And I think will be exciting years for the data regarding the PPMO and the transferrin receptor antibody, PMO, in this year. These are colleagues that are involved in the BIND project. I should acknowledge, the numbers are many, but I think some of the work that I showed from my own unit is from Raquel. I acknowledge some of the BIND collaborators when when I was presenting some of the data. The European Commission gave us money, and some of the work that I'm doing on deep phenotyping, why there is variability in individual patient receiving antisense oligonucleotide, and the culture work is partially funded by Sarepta Therapeutics. And I stayed on time, and I'll stop here.